I'm David Levine. I'm co-chair of Science Writers in New York. I want to thank Joe Bonner, who's behind the scenes producing this as always. And our guest tonight is Dr. Amit Kumar. He's the chairman, president, and CEO of Anixa Biosciences, a biotechnology company developing several programs addressing cancer and infectious disease.
Sorry, I'm having trouble. Please tell me why.
Okay. And okay, he has worked in venture capital with Oak Investment Partners, has been advisor to investment funds, venture capital firms, and Fortune 500 companies. He was on the board of directors of Acacia Research Corporation from 2002 to 2008. He has served on the board of the American Cancer Society since 2016. He received his bachelor's in chemistry from Occidental College. After graduate studies at Stanford and Caltech, he received his PhD from Caltech and followed that with a postdoctoral fellowship at Harvard. Great credentials. So today was exciting news. The first vaccines, the Pfizer vaccine was given to patients in Britain, including a 91-year-old grandmother. Was the first person. The second person was William Shakespeare. So I was just, and you said you were looking through the data, and I'm just wondering, what do you think?
Well, first of all, thank you, David, for having me. And I think this is a tremendous day, not only because of the reports of the individuals in England that started getting vaccinations, but also the data that was released, the comprehensive data that the FDA released today. And I did look through that whole report and reviewed it. And I have to say, this is an amazing vaccine. It's a hallmark day for us as scientists. And when you look at what we've accomplished in less than a year, what Pfizer and Moderna and a number of other people have accomplished in less than a year, it's fantastic. I have, 30 years ago, when I was still in school and training as a scientist, we would never have been able to develop vaccines like this that quickly. We didn't have the tools, we didn't have the knowledge. Similar to that, I look at the things that my company is doing, Anixa, and we're trying to develop breast cancer vaccines, a breast cancer vaccine and an ovarian cancer vaccine. And again, 30 years ago, we wouldn't have even contemplated trying that. But now, with the knowledge that we have today about biology, the techniques and tools that we have, we think that in a few years, we might be able to eliminate those diseases, which is, it's an amazing time to be in biotech. And the Pfizer vaccine data just supports or underscores that point.
So these vaccines are obviously giving very quick emergency use authorization. Your vaccines won't be as quick, I imagine.
Well, these vaccines are operating in a pandemic situation where people are getting sick all the time, and it's an infectious disease. And so it could quickly get out of control. In fact, it's starting to get a little bit out of control now. And so there's been an unprecedented effort by not only Pfizer and Moderna, but many, many other companies, many scientists, including my own company, to try and develop techniques, drugs, as well as vaccines to fight this. Breast cancer and ovarian cancer, while they are scourges on mankind, they are not infectious diseases. They don't cause pandemic, but if you look at the statistics, 1,700 people are dying of cancer every day in the United States, every single day that happens. And so eventually we will start focusing on that. But the research community and the media and the government has been focused on COVID for the last 10 months or so for good reasons.
So explain to the audience what emergency use authorization means, because that's not full approval.
Yeah, so EUA, Emergency Use Authorization, is a mechanism by which the government can approve certain drugs or diagnostics for use in an emergency situation, as noted by the title. In this case, the EUA still, to get that authorization, you still need to go through all of the steps that are necessary to approve a vaccine. But there are a few things that I wouldn't call them shortcuts, but there are a few things that may not be absolutely necessary. For example, typically a vaccine doesn't get approved only after 2 months of safety data. Typically, you will monitor the clinical trial participants for a year, 2 years, maybe even longer. EUA doesn't necessarily require that. The FDA is going to weigh, has probably, and is going to weigh whether the risk of the pandemic expanding is greater than the risk of not granting an authorization. And based on the data, it looks like the right thing to do is to authorize this. But we do have, the FDA does have in place, and the companies who are developing these vaccines do have in place a monitoring plan, which will monitor patients for 2 years after this initial 2-month period.
So the Pfizer's 95% efficacy is based on 170 patients. Now they had a lot more than that in trials. So how do we know it's 95% effective in?
Well, in trials like this, these are event-driven trials. These are different types of trials than drug trials where you're taking patients that are sick with some disease and you're treating them. In this case, you are giving the vaccine to a large number of healthy people and an equal or a similar number of people who, healthy people who get a placebo. And then the duration of the trial is dependent on events. And so you watch how many people in the placebo group and how many people in the vaccinated group get the disease. And so, as you indicated, it's a much smaller, 35,000, or in the case of Pfizer, 44,000 people were participants in the trial, but 100 and change got the disease. Most of them were in the placebo group. And so the efficacy is dependent on a normalized ratio of the number of people that get the disease in the placebo group relative to the vaccinated group. And so based on that those mathematics, we find that a very, very small number of people in the vaccine group got the disease, and relative to that, a larger number got the in the placebo group got the disease.
Now your company, so I want you to tell us a little bit about your company, but you're also developing a treatment for COVID-19.
That's right. So in the early days of this pandemic, some of the crystal structures, or I should say the structures of the proteins that are unique to this virus was studied and published. And we felt that there was going to be a need, and we still feel that there's a need for therapeutics that can be used outside the hospital. If you think about the therapeutics that are being used for this disease, remdesivir, dexamethasone, and the recently approved authorized monoclonal antibodies, they're all infusions, they're all injections, they all have to be used within the hospital. We felt that there was going to be a need for sort of a pill, something that if you have COVID and you start getting symptoms, you go to the pharmacy, or you go to your doctor, get a prescription, go to the pharmacy, pick up some very inexpensive pills, and start taking them before you get enough, symptomatic enough to be in the hospital. And so we decided to develop a program to identify potential molecules that can be used in that way. We've identified one that looks very promising. And as we go forward, certainly a vaccine is a great way to battle this pandemic, but we're probably going to have it's going to probably take some time to not only vaccinate the United States, but also other parts of the country. One of the things that we don't know about the Pfizer or Moderna or other vaccines is we don't know how long immunity is conferred. The efficacy is fantastic, but we don't know how long it'll last. Will it last a year? Will it last 10 years, a lifetime? We won't know those answers for quite some time. And so if the vaccine only lasts for a short duration, maybe a year or so, then there will still need to be additional vaccinations on an annual basis. And by virtue of that, we will probably still have people who become ill with COVID. And we want to be ready with a very inexpensive oral-based drug that can be utilized to outside the hospital to keep people from going to the hospital.
And what will the medicine do?
Well, the ideal situation is that this medicine will attack the virus directly. We've identified two specific proteins of the virus. One is called the main protease, and the other one is called an endoribonuclease. The technical terms are perhaps not important for our audience, but these are two specific enzymes that the virus needs to function to enable it to replicate inside the human body. And we've identified compounds that we think will inhibit or make it impossible for those enzymes to work. So these are antiviral, these molecules are antiviral molecules, similar to remdesivir, although our molecules operate on two different enzymes than remdesivir. And the goal is that these molecules will stop the virus from replicating and enable the immune system, which is already battling the virus, to destroy the virus and reduce the symptoms and eventually cure the patient of the disease.
Well, one thing you do mention is that all the drugs that we know about, such as dexamethasone and remdesivir, they were not designed for COVID-19.
That's right. So that's a good point. And if you look at the history of other types of antivirals, of which we don't have many, but the antivirals that were designed specifically against the exact enzymes and machinery, I should say, of the virus in question, those drugs tended to be better. You look at something like HIV/AIDS. Eventually, we started developing antivirals, and then eventually we developed much better antivirals once we learned more about the HIV virus. We think that's going to happen here as well. All of the other drugs, as you highlighted, were drugs that were sitting on the shelf. They were tried for other things. Remdesivir initially was tried against Ebola, but it didn't work in Ebola. And what Gilead decided was that, hey, we've got it on the shelf. We've already tested it in humans. We know it's relatively safe. Let's throw it against COVID-19. And the result was that it does have some impact. It's not a panacea. It doesn't solve all the problems and address all the issues with COVID-19, but it is something that does work to some degree. And that's the right thing to do during a pandemic. If you're in the middle of a pandemic, you look on your shelves and see what there is, and you throw everything at it and figure out empirically which of those drugs work. In our case, we said a longer term and better solution would be to start with the specific machinery of this virus, which is similar to some other viruses, similar to SARS, the SARS virus that emerged in 2003, but there are differences. And so we felt that by designing new drugs that focus on the specific machinery of this virus would eventually result in better long-term drugs. And so that's what our program is focused on.
So I want to, for the people listening, if you have questions for Dr. Kumar, and I'm sure you have lots of questions, please put them in the Q&A box, which is down the bottom of your screen, not the chat box. That's just for me and Dr. Kumar and Joe Bonner. So put them in the Q&A box and I will be happy to read them. So tell us about how you got into vaccines in the first place.
Well, vaccine technology in the history of medicine has been amazing in its ability to, address scourges of humanity. We've pretty much eliminated diseases like smallpox. 30% of the people who are inflicted by smallpox used to die. We've pretty much, not quite, but almost eliminated polio. We have now a vaccine for human papillomavirus that could potentially eliminate most forms of cervical cancer, and on and on and on. And as I said, vaccines, some vaccines have required technology that didn't exist just a few years ago. We connected with some physicians and clinical researchers at the Cleveland Clinic, which is one of the top research institutions in the world, that were developing a technique to vaccinate against cancer. Our company focuses primarily on cancer, and they had designed and developed an approach to vaccinate against breast cancer and ovarian cancer, which are two of the most common malignancies in women. We looked at that technology, and we chose to partner with them to develop those two vaccines. And frankly, the breast cancer vaccine is further along. We hope to begin human trials early next year. In the previous years, we've done a lot of animal studies showing amazing results, and we hope to be able to replicate those results in human studies. I have two daughters, college-age daughters, and breast cancer does, my family has a history of breast cancer, and I'm hoping one day my daughters will just get a shot or two, and never have to worry about breast cancer, or eventually ovarian cancer. That would be an amazing development and an amazing way to address cancer. But we've proved it in animals. We want to prove it in humans.
So I have a question. Can you possibly explain technically how a vaccine can address cancer? With a protein-based recognition system somehow not unlike what the Pfizer and Moderna strategies are.
Yeah, so a vaccine, the way a vaccine works is it takes advantage of characteristics of the human immune system. The human immune system consists primarily of white blood cells. And there are two responses that the human immune, I should say, there are two ways that the human immune system works. One is an immediate response when a pathogen or a bug enters your body. There's a brute force response that's called the innate response, and then there's an adaptive response. The adaptive response is a much more powerful response, but it takes time for it to get going. And but once it does get going, often the adaptive response has what we call memory, meaning it remembers. And if that pathogen shows up again, that adaptive response attacks that pathogen immediately before that pathogen can gain critical mass. In the breast cancer vaccine and the ovarian cancer vaccine, what the scientists, our partners at the Cleveland Clinic, learned was that there's a protein. I'll talk specifically about breast cancer right now. There's a protein called alpha-lactalbumin that is only expressed or only shows up in the mammary glands, and it only shows up during late stage pregnancy and during lactation. Once the woman stops lactating, that protein disappears. And then again, if the woman has a second child, it shows up again. That protein is important in the production of breast milk. So that's why it's needed only during the periods of time when breast milk is being produced. Now, eventually, the woman passes the age of childbirth, and that protein completely disappears. What the Cleveland Clinic researchers, led by a gentleman named Vince Tuohy, a brilliant immunologist, learned was that when a woman develops breast cancer later in life, that protein shows up again. And so his postulate was, if after the age of childbirth, 40 years or 45 years of age, we were able to teach the human immune system to attack that protein, when that protein shows up again, when the woman gets cancer, the immune system just like it would attack a pathogen, a virus, will attack that protein or the cells that are expressing that protein, which are all cancer cells. And by virtue of destroying those cells, that cancer never gains critical mass and never becomes a tumor. And so we did these studies in animals, and the initial data was amazing. We were able to show that animals that were vaccinated with the vaccine, 100% of them did not develop breast cancer, whereas the animals that were bred, these animals were bred to get breast cancer. The animals that did get the placebo, all of them developed breast cancer and eventually died. So that was a night and day type of experiment. We don't know if we'll have that 100% performance in human beings, but we're looking forward to doing that testing starting next year.
So I have a few questions about the mRNA. As a clinician, my fears are long-term adverse effects. Are there any you anticipate with this new technology, mRNA, for COVID vaccine development and manufacture? And the other question is, how does mRNA vaccines work?
So let me answer your second question first. mRNA is an information molecule. There can be trillions and trillions of different types of mRNA based on the sequence of the bases that are used to make that mRNA sequence. And our bodies, our cells, have tons and tons of different types of mRNAs that are that code for different proteins. So all of the proteins in our bodies were made by mRNA that coded from the DNA and then eventually made the protein. Those proteins are useful for our function, for structural proteins, skin, et cetera, as well as enzymes and the machinery that runs our bodies and our cells. The mRNA that Moderna and Pfizer have made comes from the sequence of the spike protein on the virus. And so the way it works is the mRNA is made, is synthesized. And by the way, there are different vaccine platforms that are being evaluated, but mRNA is the fastest, and that's why those two vaccines were first out of the gate. You can synthesize these mRNAs. They are put in a formulation that includes some fat to stabilize the mRNA for a certain degree, and then you inject them into the body. Once in the body, they get what's called phagocytosed. They get swallowed by cells in the body. Once they're in the cytoplasm of the cell, the mRNA is, the machinery of the cell takes that mRNA and makes the spike protein. And so it's just the spike protein. It's not the whole virus, just the piece of the virus. The spike protein can't get you sick, but once that spike protein is expelled from the cell, either to the surface of the cell or into the interstitial spaces between cells, that spike protein is recognized by the immune system, and the immune system mobilizes to attack that protein. So what basically you're doing is you're tricking the immune system. You're tricking the immune system into thinking that there's a viral pathogen that's in the body without making the viral pathogen. And so the immune system sees that protein and learns how to destroy it and remembers that. So now, months later or some period of time after you've gained the immunity, when the virus, when you're exposed to the virus, the adaptive immune system goes into overdrive and remembers the protein, because the spike protein is on the surface of the virus, and goes into overdrive to destroy that protein. And then eventually that virus never takes takes hold and never causes the disease for that patient. So that's how it works. The question of long-term side effects, there's no way to know the answer until you follow the patients for a long term. But based on initial data and knowledge that we have of other vaccines, most of the side effects that occur within the first couple of months. And that's one of the reasons why the FDA recently, despite objections from the administration, recently said that they want at least a minimum of 2 months of data, safety data, before they would authorize a vaccine. Now, there's no way to absolutely know. You can never say never that there's no long-term side effects, but my gut and the understanding from most of the scientists that work in this field is that there won't be long-term side effects. That being said, David, it's important for us to note that eventually, when we start vaccinating millions of people and tens of millions of people, and even eventually billions of people, there will be some people who die or have various diseases. And I can just see the headlines. So-and-so got vaccinated, and then 3 days later they died. And these are things that will have nothing to do with the vaccine. They'll have to do with the law of large numbers. There will be people that develop certain diseases, but that happens endemically in the population. So, but I can see the media turning that into into big deals.
That happened with the swine flu vaccine. A woman waited in line for 3 hours. She took a test. The next day she died, and then they looked at the number of people who would have died that day, anyways. And
yeah, that's right. So we have to understand those dynamics. But we don't feel that there will be long-term side effects. The one question that we really don't know the answer to, and I think this is an important question, is how long the immunity is conferred. Is it short-term? Is it a few months? Is it a year? Is it longer than that? The good thing that we're seeing is that over the last 8 or 10 months, during which we've been in this pandemic, we've seen a few reinfections, but not a large number. So at least we know, it sounds like we know for a period of time that lasts multiple months, we do gain decent immunity. But this virus is less than a year old, right? And the data on the vaccine is only 2 months old. And so we really won't know if we get a year of immunity or longer until we've gotten to those dates.
so your vaccines, you're testing in animals.
That's right.
Now, these vaccines are not being tested in animals. I understand it's a pandemic, but why not do tests with animals in the meantime?
We did do a lot of tests with animals.
I mean the COVID-19 vaccines.
Yeah. COVID-19 has been tested in animals. In general, FDA and regulatory agencies don't allow you to put something into a human being until you've at least tested it in animals. And the COVID-19 vaccine has been tested in small rodent type animals, anywhere from ferrets, rats, mice, and other models, and as well as non-human primates. And if there were some serious side effects that were seen in those animals, then they would not have been allowed to test in humans. So they have been tested in animals. They were just done very, very rapidly and got into human beings much more quickly than other types of drugs.
So here's a question. Where are we with our COVID-19 products? Are we in testing phase 1? Can you walk us through what steps need to get you to have one good candidate? And also, so you haven't mentioned much about Moderna's vaccine either.
Yeah, so, okay, so there are 2 questions. Let me address Moderna first. The Moderna vaccine uses a similar platform. One of the benefits, or I should say advantages of Moderna's vaccine, is that it doesn't require the storage at an incredibly cold temperature, storage and delivery at an incredibly cold temperature. The reason these mRNA vaccines require that very, very cold temperature in general is because mRNA is not a very stable molecule. Intentionally, it's designed to not be a stable, nature has designed it to not be a stable molecule. The Moderna vaccine data has not become public yet in the same way that the Pfizer vaccine data became public just today. I anticipate the Moderna vaccine will be similar to the Pfizer vaccine data, but we won't know for another week. I think they're going to release their data next week. So, it's not a long time to wait. And so we expect that to be, similar, but we'll have to see. With regard to our products, we started from scratch, and the beginning work was to look at in silico, meaning on a computer, at the enzymes that we were targeting and compare that with we compared that with over 1.2 billion molecules, which, in a way to see if the molecules would fit into the active site of these enzymes, meaning they would glum up the works of these enzymes. That process could not be done by hand. 1.2 billion molecules is a large number, but with high performance computing, you could do that very readily. And then based on that, we identified a few thousand molecules that looked interesting. Then we did some additional in silico work and culled that list down to a smaller number and brought that down to a total of 10 that we thought looked very promising. And then these are all molecules in the computer. You have to actually make them. And so we use organic synthesis methods to make those molecules. And we've made most of them. There's still more that we're planning to make, synthesize, I should say. And then the ones that we've made, we've started testing. One of them looks very, very promising. And we've done biological testing on that one and to verify that it looks promising. Now, beginning in the next month or so, we plan to do some animal tests on that molecule and other molecules that we think might be interesting. So, it's not, the process doesn't go as fast as, remdesivir, for example, where we already had lots of human data and safety data and it was sitting on the shelf. This is sort of making it from, starting from scratch. And so this is a process that will potentially take a little bit long, it'll certainly take longer. We don't know exactly how long. It all depends on the science. But we're at a stage where we have something promising. It's still early in the development phase, though.
Okay. So AstraZeneca today published an article with their data in the Lancet.
Yeah.
And so that's the first vaccine to actually have published data in the journal. What do you think of that vaccine?
So I have not read the AstraZeneca Lancet paper yet, but I will eventually. But in general, based on what I know about the AstraZeneca technology, it's also a relatively new technology. We have more history with it. That's called a vectored vaccine, where you have basically the information for the spike protein in what's known as an adenovirus, a chimpanzee adenovirus. So when you inject it into a human, our human antibodies don't attack it and destroy it. Our human immune response doesn't destroy it before it can get going. But basically, it also creates a spike protein that then gets the immune system going. The benefit of the AstraZeneca vaccine platform is that it's much more stable. And so the cold chain requirements of the Pfizer and Moderna vaccine are much less. So distribution of that vaccine is going to be easier, less expensive. For third world countries, it'll be better. The issue, though, with the AstraZeneca vaccine today is that they ended up using two dosage approaches. And one of the things that we don't understand and we'll figure out eventually is why the lower dose followed by a larger dose gave us better efficacy than two larger doses. And there's a little bit of controversy about whether that was done intentionally or whether that was done mistakenly. All of those things will get figured out eventually. And so AstraZeneca is continuing their trials, and probably in January or February, we'll see the results of their complete trial. And my gut says that they'll have similar efficacy and safety to the Moderna and Pfizer vaccines. And behind that is J&J, which is also a vectored platform. And we anticipate J&J data sometime in Q1 as well, Q1 next year. And that'll probably have similar efficacy results and safety results. And again, J&J is got the advantage of not requiring the very extreme cold requirements like the Pfizer vaccine. And J&J's design and trial utilizes a single shot as opposed to 2 shots. So we'll have to see what the data looks like there.
Okay, so someone asked, in the efficacy trials, did the investigators look at behavioral data, the risk behaviors of the placebo and vaccine groups? Do those who contract the virus exhibit greater risk behaviors for those who didn't? No, I was told that they were just told to kind of do similar things.
Yeah. So that's a really good question. People ask me why they do these massive placebo-controlled randomized trials. And the reason is you want to eliminate systemic error. So for example, if we had 10,000, 20,000 people in the vaccine arm, and a large number of them were smokers. But if we had 20,000 people in the placebo arm and very few of them were smokers, right there you've biased your result. Because if smoking is a contributing factor, then you've biased the result, and you don't get a good comparison. So you've got to have in the 2 arms, the vaccine arm and the placebo arm, similar demographics. You've got to have similar behavioral issues. You can't control the behavior of every individual in all these arms, but you want to try and randomize them in a way that you have similar characteristics and similar behavior. In the cancer arm, for example, in the cancer vaccines that we're developing, we're going to have to make sure, I use smoking as an example for COVID, but we know, and have known for many decades, that smoking is a major risk factor for cancer. So if we vaccinate a large number of women who are non-smokers with our breast cancer vaccine, and we give the placebo to a population that has a large number of smokers, chances are good that the placebo group will certainly have more incidences of cancer than the vaccine group, but that may have nothing to do with the vaccine. It may have to do simply because we didn't randomize the patient population in either case. So even though you don't necessarily tell people to change their behavior, you try, and it's never perfect, but you try to statistically double blind the people who are getting the vaccine and getting the placebo, so the individuals who got one or the other don't know what they got. The doctors who are managing those patients don't know who got the vaccine and who got the placebo. That's called double blinding, and you just have to statistically make sure that the demographics of one arm is similar to the demographics of the other.
So you're in California, lucky you, but you're also under lockdown, so not so lucky you. So the hospitals are filling up all across the country. People are getting sick all across the country, record number of testing cases. So why aren't they knocking down the doors to you and saying, you've got a treatment?
Well, eventually we will. We're still at an early stage. And eventually, once we demonstrate some animal data, which we plan to do in Q1, there's some huge opportunities. But let me make a little comment, if you'll let me, about these shutdowns. I'm sorry?
Of course.
Okay. My wife,
Yes.
I'm a scientist and I work in cancer vaccines and cancer treatment and now COVID treatment. But my wife is a physician. She's when she's on service, she sees these COVID patients, and more importantly, at her hospital, she's in administration, and she has to plan for resources because luckily her hospital is not overrun with COVID patients, but it's possible that might happen. And I can see the stress, in her and her colleagues as they deal with this situation. And frankly, I see people, and I hear on TV interviews with people who are engaging in reckless behavior. There's some simple things that we should be doing, and I'm not the only one. I don't want to repeat things that other people have said, but we should be wearing masks. We should be doing all of the things that are good guidelines and frankly, are common sense to help address this. I remember many years ago, when I was in high school, I had a teacher who told my class that when he was a kid in the, I guess the middle, 50s, he had a twin brother, and same. They both were, similar upbringing, obviously, because they were twins in the same family. But his twin brother contracted polio. Eventually, he became paralyzed and he died. He luckily did not get polio, and about a year after his twin brother became infected, a vaccine for polio was approved. Jonas Salk's vaccine was available, and he got the vaccination, and he never had to worry about polio while his brother died. And I hate to think that, and he always felt guilty and regret about that. Nothing he could have done, but he felt bad about it. And I hate to think that people who are engaging in reckless behavior today, especially when we have a vaccine that's coming in the next few days, and eventually will be available to all of us in a few months, I'd hate to think that people who are engaging in reckless behavior now, and some percentage of those people will become sick, some percentage of those sick people will go to the hospital, and some percentage of those people will die, when we can see the vaccine right there. It's in our hands in a matter of days for some people, and in a matter of months for others. So, sorry about my preaching about that, but.
So, but one thing that I heard in the news today, which is that the Trump administration they turned down by getting more vaccines, and there's a fear that we're going to run out. We're going to give out the initial doses, and Pfizer said that the next batch may not be till next summer.
Yeah, so there are 2 or 3 points that I want to make about that. First of all, I think the news came out when Scott Gottlieb did his interview, saying that Pfizer had offered this option to the administration. And I know Scott personally. Scott was on the board of directors of my previous company, so I've worked with him, and he's a straight shooter. And if he says that that's what happened, I believe him, even though some people in the administration are saying that that's not what happened.
Former FDA commissioner.
Yeah, Scott.
And he's also on the board of Pfizer now.
He's on the board of Pfizer now, but previously he was on the board of my previous company. So I know him personally. And as I said, he's a straight shooter, and if he says that was the case, despite administration officials denying it, I think that was the case. And I don't know the details of what kind of option it was. Was it just simply an option to enable more purchase, or was it a financial commitment? I don't know those details, so I don't know how and why those decisions were made. But you're right. Eventually, we will have plenty of vaccine. Eventually, initially, there will be scarcity. And in fact, there are going to be bumps in the road that are unanticipated. Already, Pfizer has said that they were planning to produce a certain number of doses by the end of the year, and they've halved that number because there are unanticipated issues regarding supply. Even in the distribution, this is a complex distribution challenge, but I believe the United States, which is the richest and most advanced technological company in the world, will figure that out. But there will be bumps on the road. There will be vaccine, shipments that'll get lost. There'll be freezers that will malfunction. And we should all just understand that that's just the nature of these types of very, very big activities. And but the key is that you may not get the vaccine immediately when you expect it, but you'll get it soon. And I'm very confident that not only will these two vaccines work, but I'm sure that the other vaccines, which have much more less stringent refrigeration and freezing requirements, and so eventually we'll be okay. But there will be bumps in the road, and again, the media will make big deals about it. But let's understand that we have done something immense, and the pandemic potentially is, the light at the end of the tunnel is there. We can see it.
How does the scientific community plan to measure the degree to which the various vaccines prevent a recipient from spreading COVID-19 to others?
So that's also a very good question. One of the answers we would like eventually, and we don't have today, is whether these vaccines simply reduce symptoms and not allow you to get sick, which is fantastic if that's all they do. But ideally, what we would like the vaccines to do is also make it, reduce infection and reduce the ability of a patient to shed virus particles. We know that there are a large number of people, especially young people, that are positive and are infected with the virus, but they're still shedding virus, and they could spread it to the high-risk population. We don't think that's the case with the vaccines, but we don't know the answer. Eventually we will, but again, as I mentioned, it's only been a few months and a couple of months with a lot of information about the vaccine. Over time, we will be able to answer those questions.
So I want to ask about the COVID-19 virus itself. We're told, wash your hands and sing along to Happy Birthday. So this virus that's killing people is pretty weak. You just wash your hands or you put Purell on it, and that kills it.
Well, yeah.
How does a virus that's so weak do so much damage?
Well, that's not the way to think about it. The virus is, so if you look at the history of different types of viruses that have been pathogenic to human beings, there are viruses that are much worse. Ebola, smallpox, as I mentioned earlier, polio, even measles can be worse. But all of those viruses can be addressed by simple techniques if we understand how they're transferred or how they're communicable. So just because you can wash your hands doesn't mean it's a weak virus. What's important is that if you get infected, that's where the virus does its damage. So if you ask me, is this virus as bad as smallpox? In the absence of a vaccine, I would say no. Smallpox is a lot worse. But if you ask me, is this virus worse than the flu? The answer is yes. It's a lot worse than the flu. So in the sort of the continuum of virus pathogenicity, this is somewhere on that continuum. It's worse than the flu. It's not as bad as smallpox. But just because you can wash your hands is not and get rid of the virus doesn't mean that it's a weak virus.
Okay, so I like to ask since we're coming close to the end, I like to ask my guest, what do you do? I mean, were you eating outdoors or indoors? Were you going to museums?
Yeah, so we, by virtue of me knowing a lot about the virus and by virtue of my wife, taking care of patients, we've been very careful. Our daughters have come back home from college. We don't travel. We have not traveled. We don't get together in large groups. We have not gone out to a restaurant, which we used to do a lot. We do get takeout food, but we mostly cook at home. And when we do meet family members or friends, we do it outside, which is difficult now because it's getting cold, but we do it outside, sitting not 6 feet apart, but even further apart than that. And so we've been very careful. And the interesting thing about that David, and we've all gotten our flu shots. The interesting thing about that is that, every year during flu season, we have I have lots of nieces and nephews who are young and who go to daycare and school, and we get together with them. Every year we pick up, common cold here and there. This year, because we've been incredibly careful due to the COVID, coronavirus, none of us has gotten any kind of common cold. But I think more important than what we do now is in the future going forward. I think there's going to be a number of people that are going to change their behavior. For example, I'm a big sports fan. I enjoy going to, football game, 49er football games and other sporting events. I enjoy going to the movies with my kids and my wife. And, in the future, I'll probably, think, I can't say never, you should never say never, but in the future, I'll probably watch, movies at home through streaming, or I'd rather maybe watch the 49ers on TV as opposed to sitting in a stadium with thousands of people all coughing and yelling and screaming, as fans. And I'm not saying that everyone will do that, but I think a number of us are going to change our behavior. I'm in my mid 50s. I'm not in the super high risk age group yet, but I'll enjoy the game from my living room, and I don't want to diminish in any way the challenges faced by businesses, restaurants, hospitality businesses, because those are serious challenges. But I don't think we should expect an immediate snapback to everything being normal. I mean, medical conferences, they're very crowded. I go to medical conferences to learn about the latest research in different fields and go see my colleagues that I went to school with. But you know what? I may not go to as many medical conferences in the future, and everyone will have to make their own decisions.
So one thing, when I was on a conference call with Dr. Fauci, he was saying what amazes him is that I think 50 to 60,000 people die a year from the flu.
Yeah.
He never wore a mask. And he said, I think people should start wearing masks during flu season. Yeah.
I mean, wearing a mask is a minor inconvenience.
Yeah.
It's not, the US, if we're asking people to wear masks, I think you said this on one of your previous podcasts, the government is not asking you to go to war. The government is just asking you to wear a mask and stay at home and watch TV. Right? I mean, it's not a great ask, and it's inconvenient. And when I wear a mask, I wear glasses. I'm not wearing them right now. Sometimes my glasses fog up, and so it's inconvenient. But it's not a big deal. It's not a great ask. It's like asking you to wear seatbelts. And as I noted earlier, the fact of the matter is, every flu season, a number of us in my family have get colds. Occasionally we get a flu, but this year none of that has happened. That's because we wear masks when we go to the grocery store. I'm in my office right now. I'm not wearing a mask, but there's no one in here. If one of my colleagues walked in, I would put on a mask. Now, I don't know if people are going to go back to, are going to go to wearing masks in the future, but you're right. Especially high risk individuals, their behavior may change, and they may be more, there may be more frequent mask wearing. That's not uncommon in other countries. So who knows what's going to happen.
In Asia. So you said you want to talk about lockdowns, but you didn't say anything about it.
Oh, well, I guess, my comment was, lockdowns, our political leaders, policymakers and executives, whether it's mayors, governors, or even the president, have been making decisions about lockdowns with very imperfect information. Now, as we've learned more, we understand that you can't completely lock down a country. You still have to have first responders. You still have to have healthcare workers, food production, et cetera, et cetera. But if you lock down on one extreme, you'll probably reduce the potential, you won't eliminate, but you'll reduce the potential for viral infections. But you'll have many second order economic effects and other healthcare effects. I mean, I know as a member of the board of directors of the American Cancer Society, there are a lot of people that are not getting cancer screenings because they're scared, and in many cases not getting treatment for their cancers, which is horrible. There are a lot of people that are not getting vaccinations for their children. I mean, it would be horrible if we have measles outbreaks, for example, in our children because of this. Or you could go to the other extreme and have no lockdowns at all, go, business as normal, and that will end up resulting in the highest number of infections. And so we have to decide, our policymakers have to decide where they want to be on that continuum. And that's a hard decision. It's very hard to criticize people that say you want to be on one extreme. It's very hard to criticize people that you want to be on the other extreme. The thing that really does bug me, though, and I assume it bugs a lot of people, is when our leaders say we're going to do X, Y, and Z, but then they flaunt those directives themselves and hypocritically do things that they've told us not to do.
Like your
Anyway, sorry, I don't want to get into politics, and that was not my intent, but you asked, so.
I didn't go to dinner with Governor Newsom, but I heard about it.
Yeah.
All right, so I have one question. I want to get to this person. What do they know about the cellular immune response to the virus? Couldn't it be possible that this is the arm of the immune response that may be more critical to defeating the virus?
Yeah, so there's what we know is that there's a cellular response and a humoral response, meaning that we see antibodies and we see T cells. So antibodies are protein molecules that are produced by a type of white blood cell called a B cell, and T cells are T cells are kind of like the stormtroopers of the immune system. They're the most powerful cytotoxic actors. They can destroy all other cells. They destroy cells that are infected, they destroy viruses, and once the T cells respond along with the antibodies, you have a very strong immune response. In the early days of the vaccines, when initially we were just looking at safety, if you recall, the news reports showed, and the published literature now shows, that virtually all of the people that got vaccinated were showing a T cell response and an antibody response. So it's hard to say which one is more powerful, but my gut says it's the T cell, but they work in concert, and it's it's synergistic.
So the holidays are coming up, Christmas, New Year's, Hanukkah, Kwanzaa, all these things.
Yeah.
Your advice not to travel, not to.
I hate to tell people what to do, and I tend to be a libertarian, and I don't like government telling people what to do and not to do. But this is a major public health issue, and to the extent that you can, try and follow the guidelines. Try not to travel. Try not to get together in large groups. If you have to, use common sense. Wear masks. Stay apart. Wash your hands, et cetera, et cetera. We just went through the Thanksgiving holidays, and we're now 2 or 3 weeks away from that. There was a lot of activity during the holidays, and we're going to start seeing how those activities impacted the surge in the viral infections. And as I said earlier, we can see light at the end of the tunnel. It'll be a shame if, because of our behavior during the holidays, that some of our family members get sick and possibly die before they get vaccinated in a matter of weeks. So think about that, but ultimately people have to make their own decisions. And I would recommend that people be much more careful than I saw during the Thanksgiving holidays.
Well, we have a governor that says, don't go visit my mom, don't kill grandma.
Yeah. Well, I mean, that's exactly right. I mean, you want to see grandma next year too, right?
Right. Yeah. Well, I want to thank you very much for spending an hour with us. And next week, I'm going to be interviewing journalists from South America and Central America and talk about their country's response to COVID, as well as they are reporting on it. We did that in Europe, and that was very successful. So I'm hoping people will tune in for that. And then after that, we're going to take a little break for the holidays. I'm not traveling, but we're taking a little break from this because it's a lot of work. But thank you so much, and I appreciate it. And if Dr. Gottlieb, if your friend wants to come on and talk to me, that would be great. Okay.
Thank you. Thank you very much, David.
Thank you. And thank you for all your work. And I hope you have a treatment for COVID-19, which would be great because we need that.
Thank you.
Okay. Take
Thank
care.
you very much. Bye-bye.
Everyone stay safe.